This grew out of a volume on dioic acid solubilities in the solubility data series. And we were keen within the SSED to explore how we could use what I guess we call more modern methods more modern methods of assessing uncertainties. And I have to say here that my expertise is considerably less than the other speakers who've been in this series. Certainly we wouldn't have achieved anything if it weren't for a great deal of input from Bryn Hibbert, who was very generous both with his time and his expertise. And probably there's nothing that I'm going to say that hasn't been covered already in the earlier talks. But what I found in doing this, and what in talking to other people, I think I'm not unique in finding, is that when you go to the guidance on reporting uncertainty in measurements, the GUM, you're confronted by high-level mathematical spaghetti, high-level equations with lots of notes telling you it doesn't always work. And it's getting from that, and it's inevitably that has to be the way it is, because the GUM applies to everything. It applies to physical measurements like the ones that we do, but it also applies to people doing nuclear physics. It applies to people doing medical studies. It applies to people doing population studies. And so the equations you're confronted with and the perspective is that of, here's something which everybody can use. And what I found was that translating that, the barrier was translating that here's something everybody can use to something I could use to solve my problem. And so what I want to do is to just track through what we did. And I'm going to de-emphasize, I'll show you the arithmetic or some of it, but I'm going to de-emphasize it because the point is that once you get to grips with it, it's actually a relatively simple procedure, or at least what we did was relatively simple. Here's a set of data we had. I've forgotten how many sets of data we had that had enough that you could analyze and try and get consensus values or best values. I've chosen this slightly at random. I wanted one with enough data you could see what was going on. One that didn't have so much data you couldn't see what was going on. This is a set of data for the solubility of pentane dioic acid in ethanoic acid. There are 5 references, and the data are here in different colors. And of course, what you need if you're going to try and assess the quality of the data is to look and say, well, okay, what's the value at some temperature? 325. Well, we've got a point here, maybe 2 points there. Orange one might be. This one's not at the same temperature. Here's a data point, but nobody else at that temperature. And so the first problem we're faced with is that the data, because of the way they've been measured, are not at systematic temperatures. They're not at the traditional 298, 303, 308 Kelvin. And part of that comes from the method of measurement, which is a turbidimetric measurement where you vary the temperature until you see the absence or presence of turbidity, depending whether you're going up and down, up or down in in temperature. Okay, so the first thing you have to do then is to bring that data together in some way. And the traditional way of dealing with these, of course, is to fit it. Sorry, I was moving all of your faces so I could see my screen. The traditional thing is to fit the data to a model equation. The Van Hoff equation is the one that's very commonly used and commonly recommended. And we did that for all, it's about 160 data sets. What we observed when we did that was that in the majority of cases, particularly where the data was quite good, you could see evidence of curvature when you looked at the residual plots. And we had a long discussion because the deviations weren't, in many cases, weren't outside of a reasonable assessment of experimental error, but they were still observable. And in the end, we went back and refitted to an extended equation. I'll come back at the end to look at that. I had an interesting email this morning from Wolfgang about whether we were overfitting the data, and I'll come back and look at that right at the end. Whoops, sorry, I've wiped out a slide. Okay, so the simplest thing you can do, and probably what lots of us would do instinctively, and probably what we would have done a long time ago as a way of treating this data. And here, by the way, I plotted the natural logarithm of the solubility. Since my variable now is the log of the solubility rather than solubility, I've plotted the log of the solubility. There's my best line through it. That's using the extended equation. It's not using it, so it's got the ln t term in it. And it reports, and just do it in ln est or in any regression program, it gives me a standard error of 0.04. And so I could take this, I could take the coefficients from this black line. I could calculate anywhere along here what the log of the solubility was, and I'd get an answer which should be good to about plus or minus 0.04. And in this case, that actually does kind of work. There's a problem with this. And whether it's a theoretical problem or a practical problem, you only know after you've gone through the analysis, because the assumption you're using when you put data into an equation is that the uncertainties, the deviations of the data are random. That is, that the data are fitting on the normal distribution curve. And that means both that the values are independent and also that the uncertainties are random. And that's not the case here. You can see this quite clearly. Here I fitted each data set to its own extended equation. Again, I've plotted the log. And now we can see that the situation is nothing like random independent data. In fact, what we have are data sets, each of which has quite good random scatter, quite small random scatter around the particular line that we put through the data. But the data sets themselves disagree. And so putting a single line through all of this, assuming that the data are random, or the errors are random, potentially is going to miss what, well, certainly misses what the real situation is. Whether that's going to be numerically important, you really have to go and check. So what you need then is a method which can take account of both systematic errors between data sets and random errors within data sets. The method chosen, and it's very commonly used, is one due to Simonian and Laird. I think the slide, I think the reference should be on the bottom of your screen. It's covered up with faces on mine. One of the nice things about the Simonian Laird method is that it's actually implemented. You can access it through websites. A very good one is the NIST consensus builder. If you have a set of data at the same temperature and you know what the deviations are or what the uncertainties are, you can simply go to the NIST consensus builder. You can put in the numbers with their uncertainties, and it'll come back and give you the consensus value. It'll give you the measure of the systematic error, and it'll give you back the uncertainty, the standard deviation in the values. Since we had quite a lot of data, we actually implemented it in Excel, and you have a copy of the Excel spreadsheet for this in that workbook which was sent round. Okay, so the Desimonean Laird method uses what I'm going to call weightings. Their symbol is W, and they're simply 1 over the square of the standard error in your experimental quantity. So what we need here is simply the standard error. Yeah, this goes all the way through these statistical things. We keep seeing the variance, which is the square of the standard error. As a physical scientist, I want the standard error because it gives me a measure of how precise the data is. But an awful lot of this is couched in terms of the variance. Okay, a small bit of, we'll get a bigger bit later, but a small bit of mathematical spaghetti. This is the thing we want. This is the variance between the data sets. It comes from an equation with 3 things in it. Well, the number of degrees of freedom is simply 1 less than the number of data sets. In this case, the number of data sets is 5, so it's 4. But we always know what the number of data sets is, so this is absolutely trivial. If we look at Q, it contains the experimental data, in our case, the log X values, but these could be enthalpies of solution. They could be equilibrium constants, rate constants, whatever they are, multiplied by the weightings. And then we have sum of the weighting times the experimental value squared, all divided by the sum of the weightings. Well, that's not too bad because we know the number of data sets, we know the experimental values, so we know all that. The W's we get from the standard deviations. So actually we know everything to do this calculation, or at least in principle we do. And the same here, our C is simply the sum. Now I've got to move you so I can see it properly. It is simply the sum of the W's for each of the data points, in this case at the same temperature. And then the sum of them squared divided by their sum. So in fact, while it looks slightly daunting when you first confront it, this is trivial. And everything else is either the experimental values or it's related to the standard deviation. Once we've got the value of tau squared, we can then go forward and calculate the consensus value. We need to define a new weighting, but it's simply the variance of the data, the square of the standard deviation, plus the variance between the data sets. So this is the standard deviation squared for the particular data point in a data set. This is the variance between the data sets. And so our consensus value is simply the sum of W star times the experimental values divided by the sum of the W star. So it's just it's a weighted average, weighted in terms of W star of the experimental values. That's not too bad. That's not difficult. And moving you again. The variance in the experimental values is simply 1 over the sum of the W star values. So if we know ln X, we know how many systems we've got, and we know the uncertainty in ln X, we've everything we need to know. We can calculate all this, get our consensus values, and we're done. Oops, sorry, I think we went through 2 slides. We do need, however, to get the standard errors, more mathematical spaghetti, but again, the point is ignore the mathematical spaghetti because it turns out that it applies to everything, but when you come to a specific case, it devolves quite simply into something that you can handle. The first thing perhaps to say is that the C here, okay, so we've got some quantity, an enthalpy, in our case, a log of solubility. and it's a function of a number of parameters. The C values in this equation are simply the differential of y with respect to the individual parameters. So C1 is dy by d beta 1, C2 is dy by d beta 2, and so on. The first term here, let me go to the next slide. The first term here includes the variance, the square of the standard error in our parameter, multiplied by the differential. So it's the sum of those for each parameter. And the second term contains the covariances. That is, how much does changing beta I affect beta j? Are they coupled in some way? In fact, what you find, there's a good example of coupling, if you look at enthalpy data or log solubility data as functional temperature with the Van Hoff equation, you find that there's quite a lot of covariance. As you add a data point, what will often happen is the intercept will change, but the B will change to compensate for it. It's quite good at calculating experimental log solubilities. It's not very good at calculating enthalpies because the slope really does depend on the intercept. They shuffle with each other. So this is a set of terms which contain the differentials times the covariances. And what we need to do is to turn this into something that is useful for our specific case. Our ln x values come from our equation a plus b upon t times ln c. And so our parameters are a, b, and c. So beta 1, let's say, is a, beta 2 is b, and beta 3 is c. So these differentials then, the c's, are simply a differential of ln x with respect to a, b, and c. I'm not going to waste a lot of time on that, specified up here, but differential of ln x with respect to a is 1. And the differential with respect to b, which is c2, is 1 upon t, and the differential with respect to c is ln t. So quite quickly we've turned the c's here from mythical symbols into concrete things that we know. And so our equation becomes 1, the differential of ln squared of ln x with respect to a is 1. So 1 times the square of the uncertainty in a, the standard deviation a. 1 upon t, these are all squared, the 1 is squared also. 1 upon t, which is differential with respect to b, 1 upon t squared times the square of the uncertainty in b, and the differential for c is ln t, so it's ln t squared. It's not a problem. And when we come down to the covariances, we just now have the differential with respect to a. This case will be the differential with respect to a, which is 1, times the differential with respect to b, which is 1 upon t, and then the covariance of a and b. The same here, the differential with respect to a multiplied by the differential with respect to c times the covariance of a and c. And finally, the product of the differentials with respect to b and c. Differential with respect to b is 1 upon t. This ln t comes from differential with respect to c, and this is the covariance of b and c. So the equation now contains things that are easy. We know the t's, so that's not a problem. And it contains the standard deviations, and it contains the covariances. Well, I'm not even going to set out to try and calculate these. This is done by, just as pH meters measure pH, computers do calculations. any regression program that you use will give you back the standard errors in A, B, and C from the regression. And many of them will give you back the covariances. We got the covariances using a covariance matrix, which is a very handy mathematical trick. And I have to thank Bryn for that. I did spend some time afterwards figuring out what it did. And I could implement it again because I have an example, so I could use it anytime I wanted. But without the example, I might struggle more than a little bit. But these things are routine. So we get these covariances from an analysis of the statistics of data. We get these. We're used to seeing these. We see these all the time. Excuse me. Drink of water. No. Okay. So let me show you what we get back, and what I've done here is to show the consensus values here, and these again. Sorry, that. Oh, ignore that 3. These are standard errors, not 3 times the standard error. I have no idea why I didn't change that. These are the data sets, and these are the standard errors on the data points from fitting just the single set of data. So this is from the regression of Song's data, this is the regression of Zhang's data, and these 3 from Liu 3 times. So these standard errors are the standard are effectively reflect the deviations from the best line through this data using this equation. Here we get the consensus value, well, not surprising. Well, I say not surprisingly, it is in fact halfway between, is where your eye or mine would put it. Here we've got quite a bit of scatter in the data. We get a 1.56. It has a standard deviation of 0.03. And it has a tau value, that is the measure of the systematic error. Let's call it systematic error because I'm old-fashioned. And let me comment, we calculated tau squared, the variance, but actually the variance tells you something about the population, which is really important in lots of uses. But for physical scientists, generally we want to, and if I measure something and I say, okay, it's 3.5 kilojoules, what I would like to know is what's the standard error in kilojoules, and I would like to know what is the systematic error in kilojoules, not in kilojoules all squared. So tau has the same units as the standard error. So it's the thing which tells you the same information that the standard error is telling you, if you like. We can see that the tau value, the interdata set deviations decrease as we go higher in temperature. And we could see that from the graph, of course. Consensus values become slightly better, they're better, and that just reflects the fact that the data are getting closer and closer together at the higher temperatures. So we've taken 5 data sets, we've come back, we've included both the experimental scatter in each of these data sets and the intersystem or the systematic error among the data sets, and that's all included here in the consensus values. So statistically, we've done, well, what I'm led to believe is the right thing. When we reported these, we actually put the consensus values through and reported consensus A, B, and C so that someone could recalculate or could calculate a consensus value at some specific temperature if they had a specific need, provided you're within the temperature range. And we did limit all of our analyses to a temperature range where there were data overlap, where there was data overlap. One slightly odd outcome, which frankly was, I think was a mistake, but it was much too late to go back and reanalyze another 160 data sets a third time. And that is that because we used the log of, well, we report log consensus value, but because we used the log of the concentration, what we got back, of course, was a standard error in the log of the solubility. And that gives you a relative error in the solubility. If I'd seen that, if I'd understood and recognized that much earlier, I would have fitted using the exponential form. So using the solubility is A plus the exponential of B over T plus T raised to the power of C, which is the exponential form of the equation we used. And that would have shifted the error onto the standard error onto the solubility. But having taken a very long time doing this and having a co-author who I'm sure who was extremely patient, but I'm sure was grinding his teeth, I didn't really feel I could go back and analyze the data. And the differences are not massive in terms of what you get. That's the output that part of the output that we provided, and it's a useful graph. The lines here are in fact the exponential of 3 times the standard error, so it corresponds to a 97 or 98 or 99% confidence limit in the log. This is the consensus value curve here. The data points are all shown, so anyone coming to the volume can look at it and say, okay, these data all fit within that. I'm quite happy with that line. One of the things that we did was we didn't exclude data points, with 3 exceptions. We didn't exclude data points just because they lay a long way off the graph. Recommendation, the normal approach now is to say, look, if you're going to put it off, you have to have some objective reason for excluding it. So the guy's thermometer didn't work, the chemicals weren't pure enough, that technique doesn't work. But just being different doesn't mean you're necessarily wrong, because what you do is you bias the population. Now, I confess there were 3 cases where I broke that rule. 2 of them, there were 2 data sets, one of which was 3 times as large as the other. So if the solubility was 0.01, in one case, it was 0.03 in another. And I simply refused to try and analyze things which are that far apart. One of them is just plain wrong. There was one set where there was good agreement between 2 data sets and a 3rd data set, which again was out by a factor of 3. And I did exclude that one. But I did say I excluded it, so anyone who wanted to include it could go back. Now, I'm not sure what is going to come up on the next slide, but yes, okay. I had a really interesting, and thank you, Wolfgang, I had a really interesting email this morning from Wolfgang Vogt saying, questioning whether we had overanalyzed the data. Now, the reason that we, as I said at the start, the reason that we went to the extra term in the equation for the log of the concentration, so we didn't stop at the BT, but we actually went out here, was that we, over 160 data sets, we could see consistent curvature. Now, consistent, so out of 160, there's probably 80, there's probably 90 had some curvature, there's probably 80 that had were good enough that the curvature was almost a smooth curve. But it was enough to convince us that it was better to include the log term. What I've done here is to put up the consensus values and the tau values. These are the ones that were on the last slide. These are the ones you get if you exclude this third term. And of course, down here where the data is pretty good, there's really no significant difference. That was true for a lot of the time. Up here, right at the end where we had those two data sets, which had values of 1.7 and 1.9, this one has given us 1.8, which is a marginally better answer. This one has given us 1.86. So the rest of these, with this term excluded, this equation was pushing the data in the direction of the 1.9 value. It's not a huge difference, but that one is now more than experimental error away from the second data point. There are only two at that concentration. So whether we had stayed with this equation or as we did, gone to the extended equation, makes very little difference. So you could argue that this is over-analysis. This one data point does argue for this. The thing is, you'd have to go back and look through the 40-odd systems that we analyzed. What you find is exactly this. Most of the time, the difference between these two is not very large. Right at the start, I showed you a graph with just a simple A plus B upon T plus C ln T equation put through it. And I said, well, it gave me back a standard deviation of 0.04. And this is what comes back from that single fit. So actually, that really simple, just put a line through the data, measure the standard deviation, assume the data points are random, ignore the fact that they're separate data sets and they're not, and the errors aren't random, you still don't get a bad answer. This one I wouldn't recommend. This one I would discuss. The problem here is that's fine for this data set where the data do agree pretty well. In some other cases, there are more data, there are data which are further off. And then this certainly would break down. Question here is still an interesting one. You want that? I said to you that on the spreadsheet, both the LinEst and also the matrix, the covariance are shown. That's just a little piece out of the spreadsheet that you got, the last sheet. There's a box that says LinEst, and these are all the way across. There's the A, B, and C coefficients. There's one called covariance, which goes all the way across. It does it for each set of data. This is just the first set. If you look at the covariance, in fact, everything you need is here. This is the square, this is the variance, it's the square of the standard deviation in A. This is the standard deviation in this is the square of the standard deviation in B. This is the square of the standard deviation in C. This is the covariance of A and B. This is the covariance of A and C. And this is the covariance of B and C. So everything you need to calculate your standard error, which allows you to calculate everything else, is in that one covariance. Okay, what I wanted to show you, and I'll stop screen sharing unless somebody wants me to, if I can figure out how to do it. I can't. This is all too complicated.
It's the same button as...
I know it is. I'm trying to get to the screen that has the button on it. I mean, you're seeing everything that's on my screen at the moment, but there's spreadsheets, there's join a meeting, we don't need that. Simao, can you simply take away the right to share my screen?
Yes, and possibly it will. Okay, that's
Perfect. Thank you. Okay, what I hoped to do, and nothing more because there's very little more I could do. What I hoped I could do was to show you that this way of analyzing data, while it's quite daunting when you come to the GUM, when you see the spaghetti arithmetic, that actually when you look at it, and particularly having an example is useful because they're kind of all the same, that you need relatively little information, and the information you need is available to you however you're handling the data. If you simply have a set of numbers at the same temperature for an enthalpy solution or something, you can go and you have the uncertainties, you can go and stick it into the NIST consensus builder and it'll give you the answer. You don't even need a spreadsheet to do that. It'll handle everything for you. If you're handling larger sets of data, the same applies. Simply, that's all the information you ever need, and it is always available to you. So people doing enthalpies as a function of concentration, or enthalpies as a function of temperature, and so on, you need, in those cases, you may well need a model equation, but then you just get the uncertainties in the parameters, and it all feeds back in. So, in fact, while I found it, personal experience, I spent the first while dutifully taking information Bryn gave me and going through it, reading the cells on spreadsheets to try to figure out what was going on. The funny thing was that eventually you got to where you knew at least what was going on, if not why, and it became a useful tool. So there, for what it's worth, there it is. And my apologies to all of you who are much better at this than I am for wasting your time. But thank you, everybody.
Thank you very much. Let's thank, even from distance, Professor Earle for giving this very nice lecture. And it was really very instructive. It was not really a loss of time. It was a gain of time for many of us. And so it is really a pleasure to listen to your presentation today. So the floor is open for some discussion, for some questions. And please just raise your hand if you want to ask something or to share some of your ideas concerning the work you have been doing within the solubility data analysis. So we have a question from Zdenek Wagner, please.
Yes, I just, do you hear me?
Yes, completely.
Okay. I just, a few minutes before starting this presentation, I calculated the same data with my tools. Can I share the screen and show it?
Yes, just a moment. It's here. Yes, please.
Okay, I spent just a few minutes by calculation, so it's not a complete analysis. And I used the same data because we used Excel files. And first I used linear regression, not in reciprocal temperature, but in temperature. And from this, and I used regression along Gnostic influence curve, which is it's inspired by robust statistics, but uses the influence function from mathematical gnostics. I will not go into equations because it will take a few hours. So it doesn't assume a normal distribution. The type of the distribution of errors is obtained from analysis of the data, and it can spread data to clusters. In the simple linear case, it doesn't show anything useful, and it shows high scatter for temperatures below 300 kelvins. So I tried quadratic regression, and it now clearly prefers Zhang's data, and just taking inspiration from the graphs in the Excel spreadsheet, I tried cubic regression. Not
And the rest of you here is Zdenek?
sorry. Do you hear me?
Yes, we are listening. Yes.
Yes.
Oh, okay. So this is a cubic polynomial in T, but not in logarithm of mole fraction, but the mole fraction. And now you see that it prefers Song's data, and it looks nice, but what should be analyzed are not the values, but the residuals. So this is the linear case, and you see that here you do not have data for zero residuals, and you see clear trends in all data sets, so the fit is not good. This is the quadratic case, so you see that the Zhang data are preferred, but still you see the trends. But so even the quadratic regression is not correct, and this is the cubic regression, and here you see that the Song's data have residuals close to zero, with probably just one outlier, and law number 3 is in a good agreement. Here you have another cluster. This is law number 1, and another cluster with a very high scatter, which is Zhang et al. So this analysis shows that there are systematic differences between probably 3 data sets, and this cluster is in a good agreement. I have also during the lecture recalculated the same with reciprocal temperature and with the equation containing the third term with logarithm of temperature. I have just the tables, not the graphs, and just looking at the tables, I think I get the similar plot as in this case. And the advantage is that I put everything together. I do not carry out 3 different fits. It finds which data match together, and there is no assumption of normal distribution, and it can find differences, detect automatically systematic differences.
Thank you for sharing your analysis over the data. Is someone else wants to do some comment? Earle, please.
Hello, thank you. Can you hear? Can you see me? No.
Yes, we can.
So thank you, Earle, for this presentation. And I have a question. So what you showed is analysis of data just as without considering the content of the paper, without analysis of the contributions from temperature effect, from impurities, from other stuff. How will it go? So how will you extend the approach you apply with involving these errors based on the experimental method. And also the second question is about the limiting behavior of the equation, because you are working close to the melting temperature of the solute. That's why it should the equation should go to the melting point of this compound with the slope equal to the enthalpy of fusion. So that's why if you include this additional constraint, it may change the form of the equation.
Thank you, Hala.
I'm not sure what, yeah, I'm not sure what the melting point is, and I confess I didn't look at that, Alice. So I don't have an answer because I don't know. It's not something I looked at. You raise an interesting question. One of the experiments, one of the methods that's used quite commonly to measure these solubilities is turbidimetric, where you take a system of known composition, well, hopefully known composition, and you vary the temperature so that, and they use laser detection. So what you see is as it goes clear, as the turbidity disappears, the signal goes up, and if you cool it back down, as you go through and it starts to become turbid, you get a fall off in the light. And so in those experiments, in fact, it's the temperature that's measured, not everything's measured, but you should be able to make up a system to a very high degree of accuracy, and the inaccuracy actually should be on the temperature. We looked at these, and what the authors do, I can't, I'm not going to second guess people. They quote their temperatures to the nearest hundredth of a degree. And if you look at the effect of a hundredth of a degree on the solubility, it's minuscule, if not zero. And so we took the decision to put the uncertainty on the effectively the uncertainty is lumped on the solubility. Now that would be perfectly good in the case where you titrate a sample of something that's been held at temperature for a long time. It's ambiguous with the turbidimetric measurements, and not having done those experiments personally, not having had hands on that instrument, I can simply go by the author's indication of the precision of their temperature measurements. So that's how we did it. That's why we did it the way we did it. When you ask the question of how would I get consensus values, taking into account both the temperature, the effective temperature and the effective and the measured solubilities.
No, I didn't mean the effect of temperature. I mean the other contributions to uncertainty, like, for example, different methods use different how to say, have different effects in the results, I would say. So impurity can affect the solubility data, for example. So the effect of impurity may be huge, especially if the sample is porous. The effect of equilibration, some methods cannot reach equilibrium. So, for example, like in this case of your case of the temperature scanning, it may superheat or supercool. It may not be the ideal. So that's why each method may have different levels of uncertainty. While you see the scatter, which is selected by the author, it's maybe not the adequate assessment of the uncertainty, I would say.
Well, yes, you're asking the classic question about post facto analysis of data, because what you have, and the best you have, is some numbers. you have what the author thinks is the purity, and in older data, you may not even have that, although quite often in older data, the authors have actually recrystallized their compound several times, so it's probably purer than some of the things that modern people use. I think all you can do with, and this isn't a counsel of despair, but I think all that you can do is to take the data you have and look to see, and the way we did it includes all of the data. You get back a consensus value. I think what's important, well, let me say something and then come back. I think what's increasingly important in analyzing data or assessing uncertainties, either in the case where you do it retrospectively or in the case of authors who are presenting new data, is that the methodology used is clearly laid out. Because then, and it was a nice example, Wolfgang wrote to me and said, I think, you know, have you overanalyzed these if we use too much equation? And the only reason he could do that was he could see exactly what we'd done. He could try it another way and go, well, and the same with Zdeněk. You know, you can look at exactly what we did and somebody says, well, I think purity has a bigger effect. But I don't know how you're going to fit that in. The problem with these, one nice thing about the turbidimetric is that at least the impurities are not insoluble ones. If there are impurities, I mean, the problem with gravimetric, for example, is that you hope there's no insoluble impurities in your compound because they're going to turn up in the solute weight. Honestly, I don't think there's a post facto way of going back to people's raw data and trying to work out anything beyond what the paper tells you. And one of the reasons for this, and you know this, and certainly over my working lifetime, if you take a particular compound and you look at the specifications and the methods of purification used by chemical companies 40 years ago or 80 years ago, and you look at the way it's done today, they're different. The minor impurities in a lot of solvents have changed over the years because the synthesis methods and the purification methods have changed. That's going to be true for solids as well. So even if you have two things with the same level of purity, 99% pure, the 1% may be different things. And without going back to catalogs and looking in what, you know, what was being sold by Merck in 1945, what their data sheets say, you've no way of even trying to extract this. And it's a problem with all measurements. I mean, calorimetric measurements, and in a way, calorimetry can be very sensitive because you're measuring, in effect, the reaction. And if you've got an impurity which reacts with your solvent or reacts with one of the other reagents in the system, it can give you typically an exothermic boost in your measured enthalpy. Even small amounts of impurities will do that. So measurements are all, and I honestly don't think, it's the reason why we left all the data in and said, look, do the statistics on the data, give the data weights which come from their internal consistency, rather than trying to, and I'm interested in what Zdeněk's seeing, because you've got two data sets which, using the Gnostics, are offline. But again, it's still not a criticism of any of these. These are all post facto, whatever set of tools you use. You can't go back to the guy's lab and say, let me see your sample, let me repeat that experiment in my lab. And you know, as I do, that even when you do round robin trials, you get different answers in different laboratories.
Earle, just one quick comment. In this case, this method will be biased towards the works which do an honest kind of smoothing of data, selecting the data which fit the equation perfectly. In this case, there will be very low uncertainty on those data. While my idea is it's better to add the nominal uncertainty for the experimental methods to each data, so you're not biased towards those unhonest.
I take that, but what I would say is that if I smooth my data, then it gets a smaller weighting. But if it's out of line with the other data, what it does is to increase the value of the tau squared. So, you know, my data will, if my data is nicely smooth and agrees with everybody else's, of course you're right, and yeah. I don't know how you deal with that. I honestly, I mean, I understand what you're saying, but so what you're basically saying is, well, look, instead of using the statistical standard deviation.
In addition to the statistical.
Well, okay, in addition to that,
the uncertainty
but could we add into that one which comes from the experimental technique?
Yes.
I guess, I mean, in principle, the answer is yes, but you need to know an awful lot more about you need to know things that you probably don't. I mean, I'll quote an example. There is, you notice there, Liu's group actually had 3 sets of data in that. I don't know why they keep measuring the same thing. And no, I don't know why. It may be that they're doing calibration and so on, but there's one case where the same group doing the same measurement with the same, well, nominally the same solvent, nominally the same solute, and nominally the same instrument have reported 2 sets of values which are 20% different. And I look at that and I go, how could you even publish that? I mean, why didn't you notice? I wish somebody would, I mean, I have lots of thoughts and something, I mean, what you say is a good idea. I just don't know how you implement it. It's where the question of having the expert doing the assessments comes. I mean, it's a good idea. It makes good sense. Authors always give you incredibly optimistic assessments of their own uncertainties and then don't tell you how they arrived at them.
So Earle, I would suggest now to listen to David Shaw. You just want to participate in this discussion, please.
Well, thank you, Samau. First, I want to thank you, Samau, for organizing this and the other meetings in this series, and you, Earle, for giving us a very interesting talk. One question that I have for you, Samau, is that you told us that the previous lectures and this one will be on the UPAC website.
Yes.
I took a moment and tried to find them, knowing that the UPAC website is notorious for its lack of search capability. And of course, I was unable. Maybe I was biased and expecting to fail.
There is no problem. We will send you the link to everyone today here in this session, and I will send the link for everybody. Hala already used her presentation in one of the conferences she attended right a few weeks ago. They are there, David.
I'm sure they're there, but they're
okay.
They're not particularly findable.
I will send
if you send me the link.
Yes,
I will put some information about this
I will
into
send
the,
to everybody that is registered for today, because everybody will can access to that. Thank you for letting me know that the IUPAC webpage is not that simple to visit and to find the things there, but we will solve it today.
thank you.
I have a question.
Okay, please, Wolfgang.
Oh, I'm interested in two things. The one thing, you have not shown the parameters itself. And all the solubilities I saw in your part 3 volume is strongly increasing with temperature. So the enthalpy of the solution should be endothermic. But in the parameters, I see that the parameters are negative, the B parameter. So it should be positive because it's minus enthalpy divided by R. What do you think about this? Second question is, I try to understand the Simonian layered formulas. And then I read through this paper. I could not find how they came to this derivation of the formula. Maybe you can recommend another source.
I confess I can't. My this I confess I took as being an established procedure, a bit like using a pH meter. And I didn't, so I made no attempt at all to try and get down inside how the black box works. And that's just a statement of fact. I didn't even attempt to do it. I was having enough difficulty converting the arithmetic into something that I could use and understanding, and then getting in the spreadsheet so they calculated what I thought they were calculating. So no, I don't, and I don't. I would, my suggestion would be to ask Bryn Hibbert.
Okay.
Bryn was, I mean, Bryn, this stuff, Bryn deals with this stuff, and it sounds really easy when Bryn's dealing with it. And it's nothing like easy when you start doing it yourself. So that would be my starting point, because he's the person, he was involved in a lot of this. He's been involved in this for a long time now. And A, he probably does understand where it comes from and so on. And B, he certainly will know the references. As to the second question,
Have you some relation to the enthalpies of the solution or
yes,
so?
no, and you're right. And I didn't even give it any thought, which for somebody who spent much of his life measuring heats of solution is an unusual thing.
Yes, this was one point, because I tried to fit the data set I sent you just to the 2-parameter equation. And so I got really an enthalpy which could be possible in the order of magnitude and also in the sign.
When you did the 2-parameter equation, the B is positive or negative?
Yes,
No, which? When you did it, which is it, positive or negative?
that's the B is negative, yes.
Yeah.
But the enthalpy is positive then.
Let me think about that. Now I'll come back to you, Wolfgang.
Maybe there are also no caloric data to compare.
I haven't looked for calorimetric data, so I don't know the answer to that. I mean, it wouldn't surprise me, but calorimetry is not a very popular technique. It's expensive and
Systems, yes.
and it's actually difficult, as for the reasons I was saying to Ala. Let me look at that and have a thought, and I'll come back to you.
Okay, thank you.
Thank you very much. Now, Alec, please.
Yes, I had a question, but before I ask, maybe a quick comment on the discussion with Wolfgang. I think when you have a 3-parameter equation, then both B and C contribute to the inferred enthalpy. So I think you'll have to just apply a thermodynamic relation to that equation and then see what it spits out as the enthalpy. But my actual question is, I looked at the method and not quite sure what it does, but my impression is that it uses the random error of each individual data set as a proxy for the systematic error. I'm wondering, is that what it does? And is that an appropriate method? Of course, if you know nothing else, then that's the only thing you can do. So that's fine with me. It's just wondering, is that what it does?
I think if you work through, no, it doesn't quite, Alec. If you look at the Q has in it the weightings times the experimental value, and then and there's a difference between, and I think that it's my instinct, because I have not worked through the, as I just said, worked through the mathematics of the method, but to me, the place where you get differences between the data sets. So I've got a sum of the variance, which is quite small for the data set, multiplied by the data set values. And I've always taken it that difference between those two terms in Q is where the difference between what's left over when you've treated it for the variance of the individual data sets. So if you, I think in that Q, that difference gives you what, in effect, gives you what's left, which you take as being the systematic error, call it. I know it's called the interlaboratory variance or something, but I think that's the term where it comes, is the difference between those two, because they have the actual experimental data in them. But you're stretching my expertise.
Thank you.